Okay, so this audio lecture will look at the slides in chapter 5, and I've broken it into two different parts. This first part is going to look at cell division and population growth. The second part, which is a separate audio lecture, will talk about environmental effects and then laboratory growth and how we actually count cells. So again, we're going to be looking at cell division and population growth for this audio lecture. So we're looking at the reproductive strategies of bacteria and archaea. So prokaryotes, most of those are going to reproduce by binary fusion. So binary fusion is kind of what we would think of like mitosis, replicate the chromosome and make sure there's enough material, cytoplasm, proteins that would be needed. Pinch in that membrane, cell wall, outer membrane, and then divide, right? So you would then have two cells. So again, we would liken that to mitosis. Binary fusion is at asexual reproduction, which bacteria and archaea do. Now there are some that will bud or have filamentous separation. So if budding, what happens is instead of a symmetric distribution of the material, one piece will have a smaller amount of cytoplasmic material. So it'll be smaller, and then that's called the bud, and the bud will go off, and then it will increase in its size. We also see this budding in some eukaryotes, like yeast. Yeast will demonstrate this kind of budding division. But it's important to keep in mind, regardless of how division happens, that they must replicate and then they will segregate their genome. And again, when we're talking about prokaryotes, we're talking about this chromosome that is circular. And so again, it would make a copy of it, as you can see in this image. So we have synthesis that occurs. So then we have two copies of that chromosome, what in this figure is referred to as chromosome 1 versus chromosome 2. We have those ribosomes, and then you will see that cell membrane starting to pinch in, and it will actually create a septum. So that septum, which is not shown in this figures, will actually be connected. We then have a pinching in of the cell wall, and there's a number of proteins and components that your textbook goes into great detail about. Some of that detail I've omitted just to make it a little more simplified, but we'll look at some of that in a little bit. So just like we as human beings and our cells have a cell cycle, bacteria's cells also have a cell cycle. So they have to go through these sequence of events in order to form the new cell. And so we can break these into three different phases. There's a period of growth after the cell is born or the last cell cycle has completed. In order to go through your complete cell cycle, you would then need to replicate the chromosomes, and then you have to partition or separate those chromosomes, and then you have cytokinesis. So again, keep in mind there's a single chromosome. Bacteria don't have the same cytoskeleton makeup that we do, eukaryotic cells do, and so those steps that we think of when a cell is dividing, it is a little bit more simpler in your prokaryotes, especially your bacteria. So when we take a closer look at chromosome replication and partition, we're going to see a figure in our next slide, but just to give you a little bit of background text on it prior, remember that most bacteria are going to have a single circular chromosome. And so they have a single origin of replication, and that's where replication is going to begin. Replication proceeds in both directions from the origin. So where it terminates, or the terminus, is going to be on the opposite side of where the site of origin begins. So here in this figure, if we look at like where 12 o'clock would be right at the top, we can see where we have our E. coli and we have our chromosome, and indicated at the very top of that chromosome is the origin of replication. So again, that is where replication is going to occur from. And then on the opposite side is where we have the terminus, and that's where replication is going to end. And as we move to like the 3 o'clock position, again, you can see that initiation of replication. And so we have the replosome that is going to bind there. That replosome contains proteins that are needed in order to replicate the DNA. And those replisomes are going to move towards that terminus. So you can see how there is that, there's an additional chromosome being synthesized. And the cell is going to have to elongate in order to accommodate this process for it to occur, which works out because we need to be able to have the double amount of cell membrane and cell wall so that we'd be able to support the two. And what you'll notice more at like the 9 o'clock position is that there's a septum that starts to form because we now have the two chromosomes that have been, one has been the template and then the other one has been replicated from that. And so again, that septum's going to form between that cell between those cells. That cell membrane again forms that septum. And then that pinching in happens, and that's because this proteins that form the Z ring pinch in on that cell wall, and so we now have cells that have divided. As just to link in with Unit 1 information, remember that when we have different arrangements of bacteria, it's due to how they end up separating. So what we're seeing with E. coli is that it's elongating and that septum forms, and it almost kind of looks like a strepto if it was to stay, but just keep in mind that some bacteria, when this replication occurs, it would be along the, if it was like a bacillus, it would be along the length of it. And so again, that septum that forms is when we're going to have cytokinesis, and this is going to allow for what's known as the cross wall to form between the two daughter cells. And again, just like our previous step had multiple steps or several steps in this process of cytokinesis. So first we have to selection of site for the septum to form. We have to have the assemble of the Z ring. This is going to be composed of proteins known as FTSZ. And then we're going to have assemble of the cell wall synthesis machinery. And so those peptidoglycan subunits are going to have to be synthesized and then assembled to form the cell wall. Then we have the construction of that septum being formed. Just to give you a visual of this process, this is not meant for you to memorize this. So this is one of these slides I'm including so we can talk about it and so you can have understanding, appreciation of it. But please do not feel like you have to memorize all of this, okay? So as you can see, especially in panel B here, you had that formation of that septum because you have these proteins that are laying the foundation, if you will, for that Z-ring formation. You should know that Z-ring formation allows for that septum for the cleaving for cytokinesis for the formation of those two cells. All right, so how does this look like when we're looking at a population of microbes? So we're going to use the term growth for the next handful of slides. And so just remember that we're not talking about an individual cell growing, we're talking about a population growing, right? So when we're talking about microbial growth, again, we're seeing an increase in the number of cells. Then we could also refer to the size, but again, what we're going to be talking about is the increase in the number of cells, indicating that there's population growth. When we look at microbial growth, we can put this onto a curve. So most microorganisms, when they're cultured in what we call like a batch culture, so when you inoculate media with a species of bacteria or from a sample, what we can do is observe their growth, their population growth, so their increase in numbers. And what we see is 5 distinct phases. And what we traditionally do is graph this. You'll notice that the x-axis is time, and then the y-axis is cells number, but log. So it is, instead of an absolute number, it's the log number of viable cells. And when we look at this, when we plot this, what you'll notice is that when you initially inoculate the media with your sample, there's what's known as a lag phase. During that lag phase, there's not a huge increase in the number of cells. So you can see how that kind of just stays the same number, a slow maybe increase. But then what we get is an exponential phase. So there's a dramatic increase in the number of cells in a relatively short period of time. And again, that would be known as the exponential phase. Just be aware if you're looking at other textbooks, they'll refer to this the log phase. And I think this textbook's really smart. They're not using the term log phase because when students write that on an exam, they'll write it kind of silly and they'll be like, I don't know which one, and it makes it grading difficult. So for our purposes, because our textbook is using lag phase and exponential phase, those are the terms you will want to use on the exam. So again, lag comes first, very slow growth. Really what's happening is the cells are adjusting to that environment. Then they've adjusted and now they can start that binary fusion. They can grow and divide, grow and divide, grow and divide. And so we have a dramatic increase in the number of cells utilizing the nutrients in the media. Again, that's exponential phase. Then what's going to happen is that population's going to kind of hit its stationary phase. Like it's not going to be able to really increase in huge amounts like it was in exponential phase. It's used up some of its nutrients. Now keep in mind these cells are undergoing metabolic pathways that can lead to the buildup of toxins. And traditionally when we're culturing them, we're not changing out and putting fresh media in during this time, right? So again, this is going to be an accumulation of some of these potential toxins into the media. There's not the replacement of the nutrients, and so it reaches a stationary phase. This is what that environment can handle for the population in that like test tube on the petri dish and such. And then eventually what happens is that there's too much toxins, there's not enough nutrients to support that population, and you'll actually see a decrease in the number of cells. And remember that this is viable cells, and so more of the cells will die, and so you see this drop of the number of cells, and that is the death phase. And what can happen is it kind of goes back down, the population size goes back down, and they can actually reach this kind of long-term stationary phase. So it's not at the high level that we were before, but there's still that population's able to maintain itself given the environment that it's in. Okay, so again, we see these 5 distinct phases. The rate in which these are going to happen differ depending upon what microorganisms are present, what media we put them into, whether that environment is ideal for them or a little less than ideal for them. So in the next set of slides, there's text of each of the phases, similar to what I just verbally went over in the figure. So with the log phase, again, this is the cell synthesizing new components. It's getting ready. It has to adjust and acclimate to that environment that it's in. So it can't just start dividing right away. It has to make sure, again, think of like all the ducks are in the row. It's doing an assessment. Like, do we have an environment that we can increase in numbers or not? Exponential phase, again, that's a term I would expect you to use on like the exam. This is where we have an increased rate of growth and division. And so we see that dramatic increase. And this is one of the reasons why the y-axis on the log scale, because if you were to do absolute number, that would increase, that it would be even more dramatic on the graphing for the exponential phase. So again, keep in mind during that exponential phase that the cells are growing as quickly as they can for those conditions. So if we were to change the conditions, if we were to change the nutrients available, that would alter that exponential growth phase and how quickly we would actually reach that stationary phase. Okay, but if there's abundance of nutrients, that's going to allow for transport mechanisms to transport that in. Eventually it gets saturated, so again, you're going to reach that stationary. They can only bring in so much material. During stationary phase, if we have a closed system, the population will end up not being able to continue in that exponential phase, and so that total number of viable cells remains consistent. Remember that there will still be cells growing and adding, but there's also going to be cells that are dying, so there becomes this balance between the reproductive rate and the death rate. So those actually become equal. So if we look at the rate of cells dividing versus the cells dying, again, during stationary phase, those are going to be equal. During the exponential phase, the reproductive rate is much higher than the death rate. Now, when we go into death phase, the death rate outpaces the reproductive rate. Okay, but again, looking at stationary phase, population may cease to divide in some cases, and they just may remain metabolically active. So there's different types of testing that you could do to determine that. So the reasons why a population might enter into stationary phase, as we've kind of touched upon, there could be limits in the nutrition available. One of the factors we'll talk about later on in part 2 of this audio lecture is going to be different factors that affect microbial growth, and one of them is going to be oxygen. So depending upon the microorganism, the amount of oxygen might limit the growth. So if there's for some organisms, if there's low amounts of oxygen, they're not going to be able to grow because they require oxygen in order to be reproductively active. We can have toxic waste accumulation. So, again, keep in mind when cells are metabolically active, they are producing potential toxins, even like CO2. And so there's a buildup of that, and that just creates an environment that's less favorable. And so, again, that would increase your death rate. And then it may be just a real estate issue. So it could be that your population density has reached critical. Like there is no more space to put any of these organisms. And if we're talking about in the lab, they're in the tissue culture dish. they're in the petri dish with their media, and there's no more surface area for them. And they've already gone up as much as they can. If we're talking about in the test tube, there's no more space for them, right? And then with death phase, as mentioned, this is where the number of viable cells declines exponentially, and so the cells are dying at a higher rate than they are reproducing, and usually there's a detrimental environmental change. So the nutrients has been used up, there's a buildup of toxic waste, and so again, it's no longer favorable for them to live. Just keep in mind that, again, for this, we're talking about like growing them in the lab, but from a, clinical standpoint, this is where a microbe might opt to go to a new location, right? So sometimes we can have an infection in one location. We're going to reach that stationary phase. Patient seems to be doing well. And then all of a sudden there's another infection. The infection is in a second location. And that is because the microbe, if it's a good pathogen, and when we talk about pathogenicity, we'll talk about how they can be motile and they can move to another location, right? So in the lab, they don't have ability to move to another test tube. But in a host, in an organism that they're infecting, they can. And then with long-term stationary phase, this is where the bacterial population might have evolved. So there's some kind of natural selection where one has certain genetic, again, mutations happen, and they may have some genetic ability to now metabolize one of the toxins. So they're able to survive. We can measure growth. So one of the common measurements we will do is calculating generation time. So generation time is the amount of time it takes for the population to double in size. And this varies depending upon the species of the microorganism and also the environmental conditions. So for some bacteria, their doubling time is 10 minutes. Others, like eukaryotic microorganisms, they can be days. And again, also depends upon the environmental condition. So we'll see in a couple slides a table from the textbook that has some different species. And one of the things I'll point out is E. coli. So E. coli has a doubling time of 20 minutes. So that means like you eat the burger that's got like 2 E. coli's on it, in 20 minutes from then, now there's 4 E. coli's. And then 20 minutes from there, there is now 8 E. coli's. And so again, it would double each time. And so within hours, you can go from having like 2 E. coli to having hundreds of E. coli. And then depending upon the species, the strain of E. coli, it might be producing some kind of toxin that could have a very ill effect on you, right? So this is why, so clinically it's important to know generation times, especially from like an epidemiology standpoint, so you can kind of trace back when the likely exposure was. Okay, and again, here on this figure, you can see how we have the minutes of incubation, and then we're looking at the red is absolute number of cells, and then the blue is log. And so this nicely demonstrates the difference between having a log scale versus absolute number. So I'm not personally going to ask you to bring in your scientific calculators and do calculations for growth rate and generation time. In the lab, you may need to do this. So you might do cell count numbers, and we'll talk about how to count these cells underneath in the lab in the second part briefly. But again, in the lab, you might have to do some of this. So just be aware on you may need to be aware on how to do calculations of growth rates, and then calculations of generation. And again, keep in mind, generation time is the doubling of the number of cells. So here you can see we've got this beginning of the growth curve for a bacteria, and there's that log phase. And then when we're in that exponential growth phase, if we look at the number of cells, and they're looking at 0.5, and this is times 10 to the 7th and 1, that is a doubling. Carry that out over to the time, and they're looking at that change of time. That would be the generation time. Okay, so that would be in the amount of time that the population doubles, and that means that a rounded division has happened. So in this table, again, I don't expect you to memorize this table, but we can, if I was to provide you with the table, I could ask questions about the table, or if I stated parts from the table, you should be able to interpret whether those were like true, false, or multiple choice type questions. But you have your different microorganisms. They indicate their incubation time, and as we will talk about in the second part, temperature makes a difference. So most microorganisms have an ideal temperature to be incubated at. And so for E. coli at 40 degrees Celsius, it has a generation time of around 21 minutes, so 0.35 hours. So again, if we had a suspected E. coli infection, we would want to go back and to think about like where did we go that we might have gotten infected with that. You'll notice that Staphylococcus aureus has a longer generation time, so they're about 30 minutes. But some of these, even bacteria, so tuberculosis, that's around 12 hours. So there's a dramatic difference in the bacteria in their generation time. And then furthermore, when we look at things like archaea and eukaryotes, their generation times can be different. And again, we can use these generation times to then indicate what, from an epidemiology standpoint. All right, so hopefully this provides you with an overview of microbial growths, how to read a growth curve for bacteria, an understanding of generation time, and then we started with looking at binary fusion. If you have any questions, please let me know, but otherwise, thank you very much.